Hello friends, this video on molecular basis of inheritance part 7 is brought to you by examfear.com. No more fear from exam. Let us now talk about the eukaryotes. So now let us see how is the DNA helix packed inside the nucleus in case of eukaryotes. Now in prokaryotes it is quite simpler when compared to that in case of eukaryotes. So what happens in eukaryotes? In eukaryotes, there are some special proteins which are present. They are positively charged and they are basic in nature. And these proteins are known as histones or histone proteins, whatever you call them. Now, these proteins are rich in amino acids and these amino acids are basic in nature. Now, as I mentioned before also, the DNA is negatively charged due to the presence of the negative phosphate group. And these proteins are positively charged. So, obviously, there will always be an affinity between the DNA and these positively charged histones. Now, due to this affinity, let us see what happens. Now, DNA will wrap around the histone octamer. What is octamer? Octa means 8. The word octa means 8. Now what happens is these histone proteins, they are like small proteins and 8 proteins group together to form a histone octamer. Now, DNA will wrap around this histone octamer and it will form a structure known as Nucleo Zone. So, you see all the terms are very similar to nucleus because they are located inside the nucleus. So, this is how a Nucleo Zone will look like. So, this entire structure is a Nucleo Zone. So, here the red colored string which you see, the double stranded structure, that is DNA. And here you can see 8 ball-like structure, spheres. Each sphere represents one histone protein. So total 8 histone proteins will group together to form the octamer and the DNA will wrap around the octamer to form a structure known as nucleosome. So this structure is nucleosome. How is this structure formed? This structure is formed because the octamer was positively charged, DNA was negatively charged, so they got attracted towards each other and that is how DNA wrapped around the octamer. Okay, so now each nucleosome contains 200 base pairs of the DNA helix. So now you see this is how it is going to be wrapping around the entire octamer. So in one octamer it is seen that the entire DNA which is present that contains around 200 base pairs. I was telling in one of the previous slides, right, that one turn or the one turn of the helix, that is the pitch of the helix, contains just 10 base pairs. So when you say 200 base pairs, that means 20 turns of the helix. So maybe like this is one turn, again this is one turn. So that is how there will be some 20 turns of the DNA which will be present in each nucleosome. Now, when we look at it on a broader level, how does it look like? So in any mammalian cell, the number of nucleosomes is equal to the number of, now this entire structure which you see here right now, it looks very big, but actually when you see it, it is a very tiny structure. So when you observe it under an electron microscope, which is a quite powerful microscope, how do they appear? You know, they will appear like beads on a string. So they will appear as small as tiny as beads. So this is how it will look like. So normally this is how the chromatin looks like, the red colored structure, right? Now if you observe it under an electron microscope, this is how it will look like. So each orange colored structure which you see, which appears like a bead, so this orange colored structure is nothing but a nucleosome. So this orange color sphere is not a single sphere, but it is a histone octamer which is being wrapped around by DNA. So this is how the appearance. And what is this red colored thread? This is nothing but the chromatin. So it will be like chromatin and in between you have the nucleosomes. So this is how DNA is packaged inside the nucleus. Now you imagine it yourself. Now if DNA is getting wrapped over something, what is happening? It is like a thread. It is a long chain. So you are actually wrapping the chain over something. So what is happening? You are actually decreasing its length. So when you wrap it over so many over so many octamers, what happens? The length reduces a lot and it becomes so compact that it fits inside the nucleus. And this is the strategy of packaging the DNA inside the nucleus. So one nucleosome contains 200 base pairs. So one nucleosome contains 200 base pairs. And I already told you that the total base pairs in a typical mammalian cell is around 6.6 into 10 to the power 9. So this is the total number of base pairs. So what do you think would be the total number of nucleosomes in a mammalian cell? So the number of nucleosomes will be equal to 6.6 into 10 to the power 9 divided by 200. So that is equal to 33 into 10 to the power 6. So these many nucleosomes will be present in a cell, and each of these nucleosomes will appear as a bit, and that is why this structure is often known as bits on string, because chromatin appears as string and the nucleosomes appear as beads. So this is how DNA is packaged inside the nucleus. And these chromatin later during cell division, they condense together to form the chromosomes. So now that we have discussed about chromatin, let us talk about a few more important things about chromatin. Now there are two forms of chromatin. One is euchromatin and the other one is heterochromatin. Now why am I suddenly discussing about the different forms of chromatin is the coiling and condensing of the chromatin actually forms the chromosome, and you all know how much important chromosome is for the process of inheritance, right? So now here we will see that during this condensing, when this condensation takes place into chromosomes, these nucleosomes, they are not present everywhere. Somewhere they are more, somewhere they are less. So that is why we are discussing about euchromatin and heterochromatin. So in euchromatin, the DNA packaging is looser. So the DNA is packaged, but it is not very compactly, or it is not very densely packed. So there are a lot of empty spaces there. So that region is known as euchromatin. So here in this picture, this is the picture of the nucleus. So where you can actually see the nucleolus. So this is the nucleolus, and here you have the euchromatin. So this portion, the yellow colored portion is euchromatin, where you just see green colored structure somewhere. It is not very dense, only somewhere you can see the green colored structure. And just outside towards the periphery, you have the heterochromatin, where you see a lot of green colored structure. So what is that green structure? That green structure represents the presence of DNA. So euchromatin has looser DNA packaging. It is lightly stained when observed under microscope. So this euchromatin, since it has lesser amount of DNA when compared to heterochromatin, so it is lightly stained when observed under microscope. So greater the amount of DNA, more will be the staining when observed under microscope. It contains less DNA. Transcriptionally active. So what do we mean by transcriptionally active? Now what happens is that for transcription, what is transcription first of all? Well, transcription is nothing but creating RNA from DNA. So I'm just giving a very simple term to explain what is transcription. So if you want to synthesize RNA from DNA, that process will be called as transcription. Now for transcription, that portion of the genes need to interact with the proteins. So in this structure, the regions are accessible. So those regions are accessible, and that is why it is transcriptionally active. But when you talk about heterochromatin, there the DNA is packaged in a very tight way. So a lot of DNA is present there. It contains more DNA. So it is darkly stained when observed under microscope. It has more DNA and it is transcriptionally inactive. That means that portion of the DNA which needs to be exposed in order to synthesize RNA, that is hidden here. Because like what happens is, if you have too much of DNA, so what happens? If it is very much compact, so what happens? That portion of the DNA which needs to be exposed for transcription, that doesn't happen, and that is why it is transcriptionally inactive. So the regions, the appropriate regions needed for transcription, they are inaccessible, and therefore transcription is inhibited. Therefore, it is said that heterochromatin is transcriptionally inactive, but euchromatin is transcriptionally active. But when you talk about the DNA content, heterochromatin has more DNA and euchromatin has less DNA. Euchromatin is found in both eukaryotes as well as prokaryotes, whereas heterochromatin is found only in eukaryotes. In prokaryotes, you do not have heterochromatin. So this was about the different forms of chromatin. Now these chromatin condense to form chromosomes during cell division. Now again, during the condensation of the chromatin to form chromosomes, who helps during that time? Again, some proteins help, and those proteins are known as the non-histone proteins or non-histone chromosome proteins. So the proteins which help the DNA to package itself, that is called the histone proteins, and the proteins which help the chromatin to condense further to form chromosomes, they are the non-histone chromosome proteins or NHC proteins. So that is also another name, NHC, non-histone chromosome proteins. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.